So good afternoon, everyone. It's a great pleasure to welcome you all to our workshop on rethinking the use of nitrogen, innovation and strategies for sustainable soil and ecosystem health. This event is part of the Open Day activities for ACTIONr, a European funded project under the Horizon 2021-2027 funding scheme. The ACTIONr project has a duration of 3 years, from November 2022 to October 2025, and we are currently in the final year of the project. And the main aim of ACTIONr is to enhance the scientific excellence and innovation capacity of the University of Thessaly by developing new tools and pathways to optimize nitrogen use efficiency, decelerate the nitrogen cycle, and reduce the environmental footprint of reactive nitrogen. To achieve these ambitious goals, UTH, the University of Thessaly, with its expertise in soil microbiology and agricultural chemistry, has partnered with two internationally renowned institutions, the University of Vienna and the group of Professor Christa Schleper, a leading group in microbial ecogenomics, and École Centrale de Lyon, and the group of Professor Graeme Nicol and Dr. Christina Hazard, known for their cutting-edge research in microbial ecology. Together, we are investigating the complex interactions between synthetic and biological nitrification inhibitors and the microbial players in the nitrogen cycle. Our research spans multiple experimental scales, from in vitro assays with individual organisms to synthetic microbial communities of nitrifiers, and ultimately to natural soil microbial assemblances. So our ultimate goals are to lower the impact of nitrogen fertilizers on groundwater and air pollution, greenhouse-driven climate change, and biodiversity loss, and of course, to stimulate public awareness about the effective and sustainable use of nitrogen. So today's workshop is a step towards this goal, and we have the privilege of hosting an exceptional lineup of speakers who will inform us and in parallel inspire us to think critically about this important topic. This workshop follows a hybrid format with some participants here in person and others join us online. So our first speaker is Professor Christa Schleper, head of the Archaea Biology and Ecogenomics Unit at the University of Vienna. She is a leading expert in microbial ecology, with her research focusing on the evolution and ecology of archaea. She is renowned for her pioneer work on uncultivated archaea in marine and terrestrial systems. Her research has significantly advanced our understanding of ammonia-oxidizing Thaumarchaeota, and in fact, we need 2 workshops just to cover the breadth of her scientific achievements. We have the honor of having her as one of the key partners in ACTIONr. And just to mention here that Christa's presentation is pre-recorded due to her simultaneously participation in a key policy meeting, but she will do her best to join us live at the end of the talk for the discussion. So without further delay, let's focus on the screen to discover what Christa has to share with us.
and how can we fix it? This is a big title. But maybe this topic also deserves big titles, because there is a big problem also about the nitrogen cycle. And I think everybody, not only scientists, but also scientists, can really fix it. And everybody can be involved. And you will see this at the end of my talk. So before I go into detail, I would like to present to you two brochures that have been written already some years ago, but they are really good, and they were written on this topic, exactly on this topic, by scientists, but meant to be for the public and for politicians, like IPCC reports. But I think these are really well understandable, and I have several slides in my presentation I have taken from these brochures. So nitrogen. Nitrogen is a chemical element. It is very important for all kinds of life on Earth. For example, or in particular, it is in every single amino acids that form the building blocks for our proteins, but it is also in our genetic material, and it is in a lot of other factors that we have in every living being. So it's a very important atom, like also oxygen and carbon and hydrogen. And the interesting thing about nitrogen is that there is a lot available on Earth, but it is in the atmosphere, as you see here on the left side. This is the D nitrogen that we have in the atmosphere. So 99.9% of all nitrogen on Earth is actually bound in the atmosphere and is not reactive. So that's the non-reactive inert nitrogen. And this is also what we breathe a lot in and out, and give it back, but it does not react really. The few nitrogen molecules that are important for all life forms are listed here on the right side with their chemical, signs. So this is nitrite, for example. It is ammonia, ammonium, and nitrate. You have heard about this. Nitric oxide is also an intermediate, and nitrous oxide is greenhouse gas. So this is also produced, and it's also a reactive nitrogen. So only these nitrogen compounds actually play a role in life on this planet. And so how do we actually get a hold of this big resource from the atmosphere of the inert nitrogen? There are possibilities. There are very strong physical forces that can bring, or that can make denitrogen to reactive nitrogen compounds. And there are also microorganisms that do biological fixation of nitrogen in legumes, in certain plants. But then, of course, we have also interfered with this nitrogen balance, if you want, between the inert nitrogen and the reactive nitrogen. And this has happened through fertilizer, through combustion, in other words, burning of fossil fuels, and then also through the cultivation of a lot of legumes, like enhancing biological fixation. And this has changed quite a bit in the nitrogen cycle. And in order to understand this or to appreciate this, I show you first the pristine nitrogen cycle as it is meant to be in pristine ecosystems. So here you have again the symbol for denitrogen in the atmosphere, and then you have the work of the bacteria that can actually fix nitrogen, certain groups of bacteria. And this nitrogen is available then for plants, for animals, every living being, fungi, everything, and also bacteria. And then there are certain bacteria that will take the remineralized ammonia that when plants degrades becomes available again. And they will oxidize it to bring it back into the overall circle of nitrogen. So this is a wonderful circle. It's like similar to the water cycle, where you have evaporation of water from the oceans, and then there are clouds and they rain down again, and the water flows into the ocean, and there's a whole cycle. This cycle is far more complex, I find. When I entered the field, I was really shocked how complex it is, but it is also very interesting. So there are bacteria that oxidize the ammonia and produce at the end nitrite and then nitrate. And nitrate is a very familiar compound for many people, because this can also wash out from the fields and end up in the groundwater, for example. And it can even be toxic. If there's too much nitrate in your groundwater, then you cannot use it anymore, the groundwater, for watering your plants, even in the garden. And also you cannot drink it then if there's too much nitrate. And we have this problem a lot. I know this well from Germany and Austria. I don't know how big it is in Greece, but I can well imagine it is also a problem. But if everything goes well, then these nitrate compounds are taken over by other microorganisms that do denitrification and bring, if they manage, bring the nitrogen eventually back to the atmosphere. And these are the denitrifiers, and intermediate compounds are, for example, for example, N2O, the greenhouse gas. So it is better if this cycle is actually well completed so that we don't have production of greenhouse gas. But now comes humans into play. Now, what happened? So with artificial fertilization, we have increased this nitrogen cycle. We have pumped in a lot of ammonia through fixation, through chemical fixation from the atmosphere. And I will come to this in more detail. And this means that all these microbes, so this cycle is only run by microorganisms, that all these microbes have more work to do actually, and a lot more. So, so we are running into trouble here because there are too many nitrogen compounds now cycling in the system of the active nitrogen compounds. And yeah, how has this all started? It is through the Haber-Bosch process. So it was Fritz Haber in 1909 who discovered the process of ammonia synthesis from atmospheric nitrogen and hydrogen. And it needs a lot of temperature, high pressure, and catalysts. So it is very energy intense. And this is why also fertilization production is one of the most energy demanding industries actually in our countries. And it was then later Bosch who developed the process to an industrial size. He was the engineer in this invention. And as you probably know, they also got the Nobel Prize for it. And the use of fertilizer has become so big that there was a comment written when there was 100 years of Haber-Bosch process celebrated. And this comment was written by Jan Willem Erisman, from whom I also have a couple of slides. And he said that Haber-Bosch are probably the most influential people among the top, 100 people or so in the world, even before Hitler and Gandhi and Einstein, etc. Why is that? What is the point? He said, Erisman said, the deliberate production and release of reactive nitrogen in the Haber-Bosch process can be considered as perhaps the greatest single experiment in global geoengineering that humans have ever made. And I would like to question this now with you. Is it really such a huge experiment that was done by bringing out reactive nitrogen to the environments? Can you really consider it such a big deal, such a big change that has happened through fertilization? And yes, there have been estimates in 2015. They will have changed now, and I'm not sure to which side, but in 2015 already, it became clear that half of the reactive nitrogen species, denoted here with NR, half of it actually stems from anthropogenic input. So here it is, the smaller part is from pollution through combustion, through fossil fuel burning and biomass burning. And most of it actually from the anthropogenic input comes from the input of Haber-Bosch nitrogen into the environments. And from these studies, it has been estimated, and also from the trend, how it is going on, or it has been going on over the past years that are not in the scheme, it has been estimated that half of humans' population would not be able to exist if we didn't have Haber-Bosch process. So without, you see a red line here, an extrapolation from the year 1900, before the Haber-Bosch process was invented. Without Haber-Bosch nitrogen, the population could have risen like this, but with this explosion, of the population, this was directly correlated to the huge input of fertilizer. And of course it was needed. It was needed to feed the world. And this was also one, or the big motivation of Haber and Bosch. Although one has to say, one motivation was also to be able to have nitroglycerin for building bombs. It was the time before the war when this investigation started. So what is the effect of this? So you could calculate, you could say, so how many nitrogen atoms do we have in our body? Yeah, if you extrapolate this on an average, every second nitrogen atom that you have is from Haber-Bosch, is from artificial fertilizer, which is probably not that evenly distributed in the world. Probably our nitrogen is probably 80% or so from artificial fertilizer. One would guess. This, in principle, is not a problem. It wouldn't be a problem. However, there is the side effect. And the side effect is so big that this is the environmental problem. In fact, most of the fertilizer does not end up in the plants. And this is the problem. It is 70%, an estimated average of 70% of fertilizer that actually is not taken up by the plants, but ends up in the environment. And this was not foreseen, of course, so it was not intentional, but it happens apparently. And I've recently seen a study, big meta-study, where they also looked at these numbers again, and they come to very similar conclusions. So on average, depending on where you live, how you do agriculture, 70% of mineral fertilizer can get lost. And the reason for that is that there are these microbes that I just showed to you earlier. Here we are showing them again, the nitrifiers, that take the ammonia, that compete with the plants for the ammonia and take it up and oxidize it. And this nitrate that is produced from this nitrification can easily be washed out because it's more soluble. And then there's also this arrow pointing to that then we have also more greenhouse gas production if the processes that should follow to bring the nitrogen back are not well in place. So this is a problem. Some people have said, we are not fertilizing the plants, we are rather fertilizing the microbes in the soil. And this is also why this workshop today is run by quite a few microbiologists, because we are caring about this, we are studying those microorganisms, and you will hear about it in the next talks. And yeah, as I said, it was not intentional, but this fertilization over 60 years now, heavy fertilization of the environment, unintentionally has caused a lot of environmental problems. And we have all seen signs of that everywhere. We can all see them. So one you maybe know is toxic algae bloomings in lakes, for example. So due to the nitrogen input, there's too many algae blooming, you know, a lot of, lot of fertilization in the lakes of organisms that you don't want to have there, for example, toxic algae. So we are here in the problem of eutrophication. There's other facets to it, a facet that rivers or lakes can also die. They lack oxygen, and a lot of organisms then die after there is too much propagation again of algae and all other kind of organisms. And then also this water, this is the area of the Gulf of Mexico, the so-called dead zone here in the Gulf, because there's a lot of towns and agriculture in this area. And this picture is not so old, it's from 2019, where a lot of fish and animals died in this zone, which was completely dead because of over-fertilization from the rivers that flowed into the environment. We don't only have this on the American continent, it's also in Europe. There's a lot of coasts you see here in red and yellow, the ones that have problems. And this picture again is a little bit older. There is a lot of investment being done now to improve coastlines, you know, from over fertilization to improve also the quality of life there again, because these ecosystems have also a big influence on tourism and so on, but also, of course, for the health of all the environments. It's very important that changes are being implemented now pretty much in Europe. The second bigger problem, apart from eutrophication, is really a sad one. And this is the problem that we also lose biodiversity by over-fertilization. So in the upper row here, you see the environment as it should be, and in the lower one is the effect of nitrogen. So you lose white flowers because only few species that grow faster and are not good in taking up nitrogen, actually, they will overgrow the other organisms. And this has a huge effect on biodiversity. Lichens are affected, that are important for the forest ecosystem. And then woodland understory here, for example, loses also biodiversity. And this is, of course, also a big effect and very harmful effect, because as you know, our ecosystems, in particular also soils, are very important for the future also. They need to be resilient, and resilience is only possible with biodiversity. And if we are losing it, we get more problems with climate change, of course, which is severe then. The third problem I would like to mention, and then I stop for the problems, I think. The third one is that the greenhouse gas N2O is also produced from the over-fertilization effect. And nitrous oxide is the third most important greenhouse gas. It also damages the ozone layer in other regions. And this should not be underestimated. The effect of nitrous oxide is so big, although there's very little in the atmosphere, but the effect is so big because it is 300 times more effective than CO2 with respect to global warming on a 100-year base projection. So 6.5% currently are estimated to be the global warming effect to be from nitrous oxide. And of course, we don't want that. So you could see this more holistic. This is done in this picture here, where you have the 5 inputs to society that are challenged by over-fertilization. One is the greenhouse balance, the greenhouse gas balance. One is soil quality. Soil acidifies with too much fertilizer. And then we have water quality, of course, air quality, and ecosystems and biodiversity. So everything is affected by nitrogen because it gets everywhere. How does that happen, and how can we also show this? I would like to show you a scheme that I find really amazing from one of these brochures that have changed my view on really the nitrogen input. I've told you a lot now, I just summarize it almost on this slide. So what you see here is the intended nitrogen flow from fertilizer and also from crops that do biological nitrification with bacteria inside their root nodules. So this is intended. We want reactive nitrogen to feed the world. And then we have the unintended, which is in greenish or yellow arrows. And that is from combustion, for example. Yeah, we also have reactive nitrogen species. And then when you go further, what happens to the fertilizer? So it should go into the crops, and then there is reactive nitrogen, and then you can feed livestock from it also, and they give nitrogen back as manure, and you can put it back on the crops, and so on. This is what we wanted. But what we really did over the past years is this one. So there's so many nitrogen species all over the place, and one single atom of nitrogen can have different effects, which is called the nitrogen cascade. So you have the leaching into the groundwater, we mentioned that. You have marine eutrophications, we mentioned that. Terrestrial over-fertilization. Then you can have gases forming N2O and NO, nitrogen oxides. Quality of air is affected, and so on. And these can be circularizing, these nitrogen compounds, unless they eventually get back to the atmosphere as N2, if they manage. And that's the nitrogen cascade, and that's the effect that we have. Here is the good one. Here's the eventual denitrification to N2. This is what we would need. Yeah, I hope that this scheme had the same effect on you as it had on me when I saw this for the first time. It is really fascinating. So then I come to a few more slides about what can we do about it? What can everyone do? Or what can society do? What can be done in agriculture? You will hear more about it in the workshop. I just touch a few things. N2O emissions, good news, N2O emissions have been reduced in Europe. Here you see the line of Europe has been reduced already through the change of agriculture. But you also see that in other continents, it's not as good yet. In East Asia, of course, is going up concomitant with more fertilizer production and more people. So there is something one can do. There is a clear effect one can do. One can reduce N2O just by changing agricultural practices, for example. And one can also reduce the ammonia input. So I give you examples here from Denmark and the Netherlands. They are really famous because they have reduced a lot nitrogen fertilizer usage and loss
Sorry about that. We will try to fix it.
Through the change of how, for example, fertilizer is brought. Compounds, unless they eventually get back. It was not possible. I have teaching duty. He's here. And of course, also we want to save CO2 footprint, a little bit to save on it. Nevertheless, I will give you like a 20-minute presentation. It is meant to be for scientists and non-scientists. So I try to get you all in the soil, just as one example. Yeah, so there's a lot of the line of Europe has been reduced already through the change of agriculture. But you also see that in other continents, it's not as good yet. And East Asia, of course, is going up concomitant with more fertilizer production and more people. So, so there is something one can do. There is a clear effect one can do. One can reduce N2O just by changing agricultural practices, for example. And one can also reduce the ammonia input. So I give you examples here from Denmark and the Netherlands. They are really famous because they have reduced a lot nitrogen fertilizer usage and loss through the change of how, for example, fertilizer is brought in the soil, just as one example. Yeah, so there's a lot of practices that can be done, and maybe it will also be discussed today in the workshop. For sure, it will be discussed what we can do with the plants. So what can everybody else do? What can we do? Like all of us, like from this moment on, we can do a lot. And this has to do, and I'm sure you have heard about it, we can do a lot by just changing our food practices. And this is not only about N2O emissions and nitrogen over fertilization. It is also about methane, yeah, because as you know, ruminants produce a lot of methane. But here we are talking about the nitrogen loss. So apparently on the way to becoming meat, this nitrogen, there is a lot of loss again. And on average, says the literature, 90% then, if you eat nitrogen in the form of meat, nitrogen is lost by 90% on the way to go there. Whereas, as we discussed in the plants, we have 50 to 80% loss depending on the practices in agriculture. So through our food choices, we can make a difference. It doesn't mean we all have to become vegan, but we can make a difference by reducing. And one of the problems why that is the case, that meat production is such a problem, certainly has to do with the fact that there's more N leakage because we have shifted from a system where local feeding inputs and nutrient cycles with the crops that we grow were close together, whereas now we have separated from each other, yeah, and do meat production in other places than where we grow crops. But to talk a little bit more about the food choice, because this is where we can really make a difference and reduce our nitrogen footprint. The WHO has reported that we are anyways taking too much nitrogen input and that it is not healthy. So by reducing meat, we can even do something for our health. And I think this is a great aspect. We can do something at the same time for the environment and for our health. And I think that many of the young generation already know this so well. It is more the generation of the boomers like me who need to learn about it. And I want to do this little sketch with you. I only have 2 or 3 minutes left, which will be enough. But I want to do this little sketch with you just for a moment, if you haven't seen it yet. What are these animals on Earth? These are earthland mammals, so no chicken. And I would like you to guess which of these groups is what. So each of the squares represents 1 million tons of biomass. So probably an elephant is 1 ton, right? We are not weighing 1 ton. So this is not weighed by individuals, but it's weighed by millions of tons. And I would like you to guess, so which group of mammals makes most of the biomass? What is the gray in the center? What is the biggest group on the left side? Maybe you stop here for a moment and make a guess. Now I show you the solution. So the cattle are the most abundant on Earth. So in terms of biomass, they are more than humans. So in the middle, you see the dark gray, of course, it's anthropocentric, this picture. In the middle, you see humans represented. And the elephants are down here, this one little green dot. And then you see other animals, of course, they are all for meat production in the world, all the big groups that we have. So we have really changed all ecosystems. No wonder that there is a problem. So this is the size of the topic, right, that we have to deal with when we produce meat. And this is my last slide to hand over actually to the next talks. Another thing that can help, of course, to change the nitrogen problem, to help the nitrogen problem, is change in agriculture. And this will be addressed in the next talks. And there's a lot of opportunities and possibilities, and already a lot of action is being done, fortunately. And I'm pretty sure that this will change a lot. So with that, I would like to end. I thank again all people from the ACTIONr network, also the colleagues in France, but particularly in Larissa. Thank you for being there today. And I thank also my group for all the excellent work that they have done on ammonia oxidizing archaea over the past 20 years. Thank you very much.
Christa, thank you very much for your inspiring presentation. Are you here with us? We can see you.
Yes, I'm there. I'm a little bit in the dark, and I'm in a special place, you know, on a meeting, but I would answer questions if you want, if you can hear me.
We can, yes, of course. Are there any questions from our audience? Now it's your opportunity to discuss with Christa Schleper. Go ahead.
First of all, where are you, Christa?
Oh, I'm in a cafe and I'm in a highly political discussion for the Senate of the University of Vienna.
Oh.
We are forming ourselves as a political force. And that's why there's such a background, you know, around the corner, because it's in a back room of a cafe.
That's nice. Okay, so any questions for Christa? Come on.
Maybe most people knew what I was talking about, right?
Yep. Come on. Questions? They are shy. Now Poppy has a question.
Hi, Christa. This is Poppy.
Hello.
Are you a vegetarian?
I am mostly a vegetarian, yes. Sometimes I eat meat because I cannot stand milk products. I have to eat meat to get some food in some places. You know, when I have choice, I am vegetarian or vegan. Are you?
No.
I don't
But
think
now
we
I
have
will,
all,
I
we
will
all
start
have to
thinking.
become.
Yeah, I will start thinking about it now.
Yeah. I don't think we all have to become very strict vegetarians, you know. If we just ate meat like we did in our childhood, you know, in my age 50 years ago, then it would also be okay.
Yeah, this is
It would
true.
be different. But we organized vegan scientific meetings in Vienna. Graeme remembers, right? And people liked it. They had curry every day.
It's true. It was good.
It was something new. But you cannot organize a nitrogen meeting and have meat for food. That is really a little bit odd. Okay, then I wish you a very successful continuation. Maybe I will join in later in about an hour or so and listen to the other talks. I will miss you, Graeme. I'm sorry for that. Now you guys get to the details.
That's okay.
Is there no more questions? Okay, thank you.
Okay, so next we have Professor Graeme Nicol. Graeme is a director of research at the French National Center for Scientific Research and a member of the Environmental Microbial Genomics Group at École Centrale de Lyon. His research has revolutionized our understanding of nitrogen cycling in soil. His team made history by cultivating Nitrosotalea devanatera, the world's first obligate acidophilic ammonia oxidizer, solving the long-standing puzzle of nitrification in acidic soils. From pioneer stable isotope techniques to exploring the hidden world of soil viruses, Graeme's work continues to illuminate the complex interplay of microbes and biochemical processes. Graeme, welcome. The screen is yours.
Thank you so much. Can I just confirm that you can hear me?
Yes, we can hear you, Graeme.
Okay, great, super. Microsoft Teams would like to record this. Yeah, that's fine. Yes. Oh, sorry, I have to give permissions for Teams. Excuse me. I'll be back. I have to quit and reopen Teams so I can share my screen. Excuse me.
Stay with us.
And he's back. Okay. Share my screen. Okay. Sorry for that little delay. So first of all, it's
Graeme?
everyone. Oh yes.
Okay. Yeah, we can see your presentation now. Go ahead.
Great. Okay. Thanks. So, so first of all, good evening, everyone. I hope you've had an interesting day at the workshop, and following on from Christa's talk, which is always very inspiring. It's always nice to see her, although it's always a little daunting to follow her giving a talk, because it's just very good. I'm going to talk a bit more about the microbiology that's underpinning the processes that Christa was talking about, and what we in Lyon with our colleagues in Thessaly, at the University of Thessaly and at the University of Vienna are working on. So the title of my talk is Why Understanding Soil Microbiology is Essential for Mitigating Environmental and Economic Impacts of Fertilizer Use. So just a little bit of revision from Christa's previous slide, but the point of this slide is, again, just to reiterate that artificial inorganic fertilizer is responsible for feeding half of humanity. And so the Haber-Bosch process, as Christa mentioned, it was invented by Fritz Haber and industrialized by Karl Bosch, and they both got different Nobel Prizes for this process, but they're responsible, this process is responsible for generating the fertilizer which feeds half of humanity. An important point is that not only is it responsible for generating huge quantities of fixed inorganic nitrogen, but it actually has a very large carbon footprint. In fact, it's estimated that around 2% of all global energy demand is actually used for the production of ammonium using this process. So not only is it accelerating the nitrogen cycle, but it has a very important impact on the carbon cycle as well. And again, in terms of it's why we're particularly interested in the issues surrounding the use of nitrogen fertilizers, as Christa mentioned before. One of the problems is the production of nitrous oxide. So again, the third most important greenhouse gas. And unlike, so after CO2, which is the one that we're usually most concerned about, and then methane, it has the third amount of radiative force compared to other gases. Unlike carbon dioxide and methane, it has a much longer half-life in the atmosphere. So that's why we're particularly concerned about it. And in terms of its sources on the Earth, soil is the largest source of N2O. And within soil systems, it's actually the use of the addition of these artificial fertilizers, which is really accelerating this increase in nitrous oxide emissions since the beginning of the Industrial Revolution in the mid-18th century. It's also primarily it's considered to be or predicted to be the primary depleter of ozone in the 21st century. So artificial fertilizer has accelerated the natural nitrogen cycle. So we saw a few variations of the concept I'm about to cover, but this is sort of a simplified version. So we can all understand that we're all very familiar that plants that feeds animals and humans directly through eating crops. We all understand that these are photosynthetic organisms and they're fixing carbon dioxide from the atmosphere. But in terms of the nitrogen, plants themselves cannot take nitrogen from the atmosphere. So nitrogen in the atmosphere is around 70% of all the gases, and it's very chemically inert. So it's very difficult to react with. And therefore, plants and animals can't take nitrogen directly, and we're relying on many different mechanisms, but the one that we're mostly concerned with is the activity of microorganisms in soil systems, where they take nitrogen from the atmosphere and they turn that into ammonium. And so this is the process of nitrogen fixation, and it can be performed by free-living bacteria, which are resident in the soil, but a lot of the time it's done by bacteria which live in nodules inside rhizobial plants, so things like clover or beans, for example. These are small sort of organelles and plant roots which are responsible for high rates of activity where nitrogen is coming from the atmosphere and being fixed into plants and into the soil systems. So, for example, this is why we use farmers will sometimes will plant clover after a cropping of a different arable crop, because the clover has these nodules with the nitrogen-fixing bacteria, and it's a way of replenishing nitrogen in the soil that has been used by previous cropping systems. So nitrogen fixation, natural process, and is responsible for around approximately 80 billion kilograms of nitrogen per year. Now, because we have such high demands for crops for feeding ourselves directly and also for feeding animals for consumption, we're using these artificially produced inorganic fertilizers to essentially bypass the natural nitrogen cycle. And what's quite astounding is that we always think of carbon as the carbon cycle as being the one that's most impacted by humanity, but I think you could argue strongly that it's actually the nitrogen cycle which has been on a sort of on a relativistic basis. It's actually the nitrogen cycle which has been much more impacted by the activity of humans. So we actually have 150 billion kilograms of nitrogen going into the soils, which is fixed by the Haber-Bosch process. So 150 billion kilograms of nitrogen is it's quite difficult to visualize. It's quite a huge amount, but maybe, I don't know if there's an easier way, but a statistic is that it's the equivalent of 24 million elephants of nitrogen is added to the world's soil. So following on from Christa's talk, she was talking about elephants, and so I've tried to follow that process by converting nitrogen into elephants. But anyway, 24 million elephants is still quite an unimaginable amount. But so when we're adding this nitrogen into the soil as fertilizer, as ammonium, what we're trying to do is we're trying to add nitrogen into the soil, and therefore arable crops can take up the nitrogen and have higher yields, because they can take the carbon from carbon dioxide from the atmosphere, but it's really nitrogen which is one of the major limiting nutrients. So that's why we add inorganic nitrogen fertilizer. But what actually is happening is that the nitrogen that we add into the soil, the majority of that nitrogen is not being taken up by plants. But there's actually a competition, as it were, between the plant's ability to take up that nitrogen and also microbial processes which actually convert that ammonium through the process of nitrification, which results in the loss of the nitrogen and its unavailability for the plants to take it up. And therefore, this reduces nitrogen use efficiency. And then this is one of the main concerns for nitrogen pollution. So the process of nitrification is the oxidation of ammonia, the most reduced form of nitrogen, all the way through to nitrate, which is NO3. And then these steps, the oxidation of ammonia through to nitrite in the middle, and nitrate, NO3, on the right-hand side, is all performed by microorganisms in soil. And this is a natural process. These are organisms which are naturally current on soils, current in different environments, marine water, fresh water. They're ubiquitous in nature. But one of the consequences is that because we're accelerating this process by putting all this huge amounts of nitrogen into soil systems, we're also increasing the rates of production of certain compounds which doesn't have a chance to be incorporated back into the nitrogen cycle in a sort of natural process. And so one of the main consequences is the production of nitrate, which is leached into waterways, and that leads to eutrophication. So there's a picture there of Erie Lake in the US, where you have algal blooms because there's just so much nitrogen there, it's just alleviating restrictions on the growth of the algae. And then when that happens, you get huge amounts of biomass of algae, which dies and sinks and depletes all the oxygen in these systems when the algae is actually getting degraded, and therefore that's when you get these pictures of fish dying when there's algal blooms, because there's no oxygen left in the water. And then, of course, the other consequence which we've been talking about is the production of nitrous oxide production. And so one process in a natural part of the nitrogen cycle is denitrification, where we're taking nitrate, NO3, the most reduced form, and we're returning that back to the atmosphere. But because we're accelerating it so much, this process, we're just getting huge amounts of N2O being produced. And we're not really giving it enough, we're not giving it the opportunity to be turned back into the dinitrogen. So just to talk about the sort of the fundamental work that we're performing in the ACTIONr network is really based on trying to understand how microorganisms work in soil. So For those of you who are not soil microbiologists, it might surprise to know that the soil is actually probably the most diverse, biologically diverse habitat on the planet. So you can actually call it the rainforest beneath your feet. So if you think of this spoon as quite a large spoon, it has 10 grams of soil. This will contain about 10 to 100 billion different bacterial cells. And this will represent over 1 million species. And one way to think about that is that the amount of diversity, the huge amount of diversity of bacteria species in soil is almost equivalent to all the different species of higher animals like plants, animals, insects, fungi, all those different species that we see every day in our lives. That total number is actually approximately the same, or maybe even slightly less, than just the number of different species of bacteria that we find in just one large spoonful of soil. Now, that's really quite an amazing fact, which we've been trying to understand for the last 20 to 30 years. But one of the major problems is that we can't cultivate these organisms in the lab, and it's very difficult to understand what all these vast diversity of different species of bacteria are performing in the soil. And therefore, at fundamental level, we are still characterizing microbial populations that carry out fundamental processes, such as nitrification, at quite a basic level. So we haven't even solved understanding all the different organisms that are performing these processes. So if we go back to nitrification, and this is the process where we're oxidizing ammonia through to nitrate. Again, this is the ammonia could come from added fertilizer. There's been a history of the discovery of organisms and understanding those organisms which are performing this process. And this happened in the late 19th century by scientists such as the Faraday Franklins in Scotland or Winogradsky in Russia. And then they found that there's different groups of organisms, ammonia oxidizing bacteria, which we call AOB, and these are organisms which do the first step, oxidizing ammonia through to nitrate. Then there are other organisms which are specifically adapted to only oxidizing nitrite through to nitrate, and they're called the nitrite oxidizing bacteria. And then around 20 years ago, we had the discovery of ammonia oxidizing archaea, and actually Christa, who was in the previous talk, she was actually very fundamental in discovering these organisms that perform this process. And then a little bit more recently, there's another group of organisms, again, also found in soil, which can actually perform this entire process of nitrification, not in 2 steps, but in 1 step. They're complete ammonia oxidizers, and we call them collomox. So these are different organisms, and we've got their scientific names here, and it's not really important, the details, but what I'm trying to show here is that there's a diversity of different organisms that perform this one process. And we're trying to understand the reasons why there are a range of different organisms that perform this process, and what are the consequences for actually nitrogen cycling, and particularly what are the consequences of this diversity for understanding how nitrogen fertilizer is transformed from ammonia through to nitrate in soil. So this is just one slide which sort of summarizes quite a lot of work, but in terms of the laboratory experiments that we perform, we're trying to understand how these different groups use nitrogen in soil, and how these different groups actually produce N2O. And through many, many experiments over many years, what we've actually found is that these different nitrifier groups that we find in soil actually have quite different physiologies. And what they're actually able to do is some will use fertilizer more than others. And subsequently, some will also produce more nitrous oxide than others. So we can also think of some groups of nitrifiers or populations or communities that we really have to worry about and we have to be concerned about. And then there are other populations and communities which we can be less concerned about because, one, they're not really utilizing the fertilizers that we add to the soil, but they're also not producing high yields of nitrous oxide. And in particular, if you can remember some of those names I presented in the last slide, we have ammonia oxidizing bacteria. These are the organisms which have been known about for over 100 years, almost 150 years now. And these organisms are the organisms which are using nitrogen in fertilized soil. So these are the organisms which use and transform and compete with plants for added ammonium fertilizer. So these are the ones that we have to worry about. Other populations, such as the ammonia oxidizing archaea, the ones that we've known about for only about 20 years or so, they seem to be more important in unfertilized soils. So where the nitrogen cycle, you can think of it's happening under more natural conditions, where there's not huge amounts of nitrogen fertilizer being dumped on the soil, there's not huge amounts of nitrous oxide being produced. And then so in the unfertilized soils, it's the ammonia oxidizing archaea which are less of a problem. So if we take this back to the field and we think about how can this inform how we manage our fertilizing strategies and how can we try and reduce our the pollution that's produced from adding fertilizer to soil, we can think about the physiology of these different populations. So here in this cartoon, we have the ammonium-oxidizing archaea. In the purple, we have the ammonium-oxidizing bacteria and these sort of spiral-shaped cells. And based on the experiments that we've performed, not only us, but in many different labs, what we've discovered is that when we add inorganic ammonium fertilizers to soil, we get growth of these ammonium-oxidizing bacteria, the populations that we have to worry about. And that's because even if we just add a single application of fertilizer, we get huge increases in the growth of these populations. And associated with that, we get large amounts of nitrous oxide being produced. So this is the troublesome part, because it's these organisms which are responsible for removing the ammonium fertilizer away from plants, but also converting that to more polluting forms. If we think about the archaea, the ammonium-oxidizing archaea, these ones preferentially, well, these ones do not utilize your standard ammonium fertilizers produced through the Haber-Bosch process. But what we've discovered is that they can use more organic sources of nitrogen or ammonium that's added at very slow rates. So when you're adding ammonium at a slow rate, it has two advantages. One, the ammonium-oxidizing bacteria can't use it and produce large amounts of nitrous oxide. But secondly, they can't, they don't produce high rates of nitrous oxide. So even when they are active and when they're dominating activity, they're much less of a problem than the ammonium-oxidizing bacteria. So that's sort of the fundamental discoveries that we've made over the last sort of 10 years regarding the physiology of these different microbes in soil and their contributions to nitrous oxide. So this is just my last slide. So how does this knowledge of ammonium oxidizing physiology and understanding these different populations that find in soil, how can that be used to inform mitigation strategies? So this is just an example. So again, we've got this cartoon here where we're adding fertilizer to the soil. And when we add ammonium, what we're wanting to do, we're wanting it to go to the left, to go to the plant roots so it can take up nitrogen and increase the yields of crop plants. But actually what's happening is that it's actually the majority of it, 70% of the nitrogen that we add to soil is not going to the plants, but is being utilized by ammonia oxidizing microorganisms. And that subsequently fuels these processes of denitrification and nitrification, which results in the production of nitrates, which are leached into waterways, and also the production of nitrous oxide. And so what we're trying to do is we're trying to think of strategies where we can almost control these populations in soil. Remember, these organisms are naturally occurring in soil, and they perform the natural process of nitrification, which is essential to the global nitrogen cycle. But we're actually wanting to reduce their activity in situations where we're adding quantities of fertilizer for increasing plant yields. So one option is to perhaps use slow-release fertilizers. Again, adding ammonium and slowly increases the chances of the plant taking up the ammonia, but also removes the ammonium from the pathway that feeds the ammonium oxidizing bacteria. And then leading to the next talk from Professor Karpouzas, we're also investigating the use of nitrifier inhibitors. So you can add these to soil at the same time, and that will hopefully target the nitrifier specifically, and that will reduce their activity under circumstances where we're adding fertilizer. So that's really sort of where we're trying to be informed by the physiology of microorganisms and trying and reduce the use of try and reduce the effects of adding fertilizers. Okay, so that's just the end of my talk. Just to acknowledge the funders. Again, this is our research group here. It's actually actually the lab meal from last night, so it's quite a nice photograph. So I thought I would utilize that today. And then, of course, I'd like to thank ACTIONr and the European Commission for funding this very important research work. And thank you for listening.
Thank you, Graeme, for your talk. Do we have any questions for Graeme?
So, as you said, a lot of the nitrogen we use in fertilizers is in the end wasted, so that a lot of the nitrogen doesn't reach the plants. But we seem to be so afraid to do away with the usage of fertilizers. So, even in the last slide, you said that we're trying to manipulate the microbial populations to control the dosage of the fertilizers to the plants and how the microbiome reacts to this. Is there any hope that this manipulation of the microbiome could support plant growth without fertilizers, or should we still try to make the use of the fertilizers that we're making more sustainable?
That's an excellent question. I'm not an agronomist or a econometricist, but I'm quite confident that we wouldn't be able to sustain current yields of crops by not having any form of fertilization. By manipulating or controlling the nitrifier communities, however, what we do is we do have an opportunity, I think, and a realistic opportunity to reduce hugely the amount of actual fertilizers which are added. So it's not just a matter of adding the same amount of fertilizer and adding compounds or using strategies which inhibit the microbial activity. By increasing this nitrogen use efficiency, we could imagine scenarios where we can actually apply just 1/3 of the nitrogen fertilizer that we're used to, because 70% or 2/3 would get lost. So I think I think we'll always require fertilizers, but I think we have realistic opportunities to really reduce dramatically the amount of fertilizer that's added, and that also has huge economic benefits as well.
Thank you. But even about the slight manipulation for manipulating the response to the fertilizers, are we sure about the long-term effects that is going to have to the soil health? Are we sure that we're not playing in some ways that we're going to regret later?
It depends on how you're manipulating it. If you're using nitrifier inhibitors, I think that's a very important question, and that's actually one of the topics that we're trying to evaluate in the ACTIONr program. These organisms are natural, and by reducing their activity, we're never going to. I think it's unlikely that we're going to have like a diversity effect, like we're reducing biodiversity when we're concerned about the Amazon rainforests, etc. But what we do have, what is an important consideration, is what's the impact of these scenarios on other organisms. So if we're adding sort of synthetic chemicals to inhibit nitrifiers, I think it's a very good chance that it's not just nitrifiers that are affecting, and then you're reducing lots of other you're reducing the activity of lots of other microorganisms which are performing intrinsic and fundamental and important processes for soil health. So I think it's specifically for the inhibitor aspects. I think it's something that we have to be very careful of. Yes.
Thank you very much.
Are there any other questions, Graeme, or comments maybe?
So, Graeme, do you want to say a few words about your phage therapy approach? Because we are coming from a department that we have a lot of background. I mean, our students have a lot of background knowledge on medical issues. So phage therapy is quite I would say well-known approach to tackle a lot of medical issues. But I find your idea of phage therapy nitrification quite fascinating. So you want to say a couple of words about it?
Yeah, I would like to. Thank you. So so our maybe a polite word is ambitious research program. One of our okay.
I think it's
okay. They're remuted now. So so one of the concepts, again, coming from the medical field, is the application of viruses, which lytic viruses specifically, which infect your target population and reducing their activity. And so when people have, for example, like a skin infection which can't be treated by antibiotics, sometimes the use of a phage, a virus which is very specifically targeting a Pseudomonas, for example, can be used. And so the concept for the phage therapy for soils is that a natural and highly targeted approach, instead of using chemicals, would be to cultivate, produce and apply viruses, phage, which target only the nitrifiers which are performing the process that we're trying to eliminate. So that would be a very highly targeted approach. It would be natural because we're not adding any chemicals, and we're only using viruses which naturally occur in soils or other environments. And so that could be applied, the viruses could be applied at the time just when there is a fertilization event. And I'd be knocking down, reducing their activity.
Excellent. So any more questions for Graeme? Okay. So Graeme, you're going to be around?
Absolutely.
Right. Okay. Thanks.
Okay, thank you.
I need to be introduced.
So our next speaker is Professor Dimitrios Karpouzas, head of the Department of Biochemistry and Biotechnology, and of course the coordinator of the ACTIONr project. He's a leading expert in interactions of agrochemicals with soil microbiota and has extensive experience in developing biotechnological solutions to improve soil health and reduce environmental impacts. Especially today, at the end of his talk, he will be awarding an Oscar. So let's find out who the lucky winner is.
Okay. 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 So, okay. So, good afternoon. So indeed, I will try to give an Oscar. So I'm going to give you an insight on what synthetic chemists can do and what plants can do. And there are some biased people in the audience, like Professor Papadopoulou, that I'm pretty sure before the start of my talk will vote for plants. But we are going to answer the question: who's the best in putting the brakes on soil nitrification? And if the approaches that synthetic chemists are following are more efficient, and I would say more efficient, not just to decelerate nitrification, but also if they are actually sustainable ways to decelerate nitrification. So certainly synthetic chemists can synthesize a lot of really interesting chemicals that we are all using in our everyday life. So one of these examples is benzimidazole derivatives, that they are pesticides, they are veterinary medicines, and they are a lot of these chemicals that we are using in our everyday life when we are having some infection or whatever. And if we look up a bit more in the synthesis of chemicals that they are actually useful in the nitrogen process, they can certainly synthesize triazole derivatives. Triazole is a skeleton of a lot of synthetic nitrification inhibitors that you've heard in the previous two talks. So triazoles have been used as agrochemicals, as human medicines. So chemists generally can do a lot of nice things. They can synthesize a lot of nice products. But this is a genuine discussion I had with a synthetic chemist that we have in our department. The students that they are in our department have they know him, Professor Komniotis. Okay, so this is a picture that was taken 14 years ago in the celebration of the first 10 years of the department. But this is a genuine discussion, I tell you, okay? I'm not joking. So I asked him, we were sitting there, and I had a talk after that discussion, and I was presenting how microbes can actually serve the environment and be the cleaning up ladies of the environment, so it can clean up the mess that we are actually creating. And I asked him, I gave him a piece of paper with a metabolic pathway that we had with a pesticide that our microbes were able to degrade, and they were producing a lot of different intermediates. So I gave it to him and I said, can you do something about? Can you help? Can you actually synthesize all these some of these metabolites so we can play around and see if they are produced or if they are not produced? He looked at it, took him 1 minute or so, and he says, no, I cannot do much about it, because what microbes can do in 24 hours, it will take me 3 or 4 years to actually synthesize those things. So I was, on the one hand, I was disappointed because my brilliant idea didn't go forward. But on the other hand, I was a bit convinced that I'm on the right path, working with microbes and not becoming a synthetic chemist. So these are some of the molecules that we were not able to synthesize in the bench, but microbes were very nicely producing all these nice intermediates. So what plants can do, on the other hand? Plants can synthesize and exude molecules, and these molecules serve different purposes. They defend against enemies. They communicate with other entities in their vicinity. They might be friends or they might be enemies. They modulate their growth by exudating stuff in the rhizosphere. They enhance their capacity to acquire nutrients from soil. But overall, what they do by exudating all these molecules in their rhizosphere, in the ball of soil that is adjacent to the plant roots, they modulate, they create a microenvironment that suits them better. So what do they produce? What is all these exudates that we are talking about? They are all sorts of different things. They are amino acids, they are organic acids, peptides, hormones, phenolic acids, coumarin, sugars, whatever you call it, flavonoids, fatty acids, sterols, all sorts of different things. And they are doing all these really nice services that I've explained before. However, the key element of what the synthetic chemists can do and what the plants can produce is how they fit all this stuff in the nitrification process. And you had two experts before me that they described very well the nitrification process. And as Graeme introduced very nicely, there are a lot of different processes that they lead to undesirable products that create a lot of environmental concerns, like the emissions of nitrous oxide, or the production and accumulation of nitrate that can leach into the groundwater. So all these processes makes us think how we can decelerate nitrification that has been enhanced by our anthropogenic activities, by putting a lot of surplus of nitrogen and trying to actually push more and more. And this has created all these problems. And we have, on the one hand, our synthetic weapons that we can use, like synthetic nitrification inhibitors, that we can use to decelerate nitrification. And this is not a very rich arsenal of products that we have. Consider that we have now 3 or 4 cascades of chemicals that they are sold as synthetic nitrification inhibitors. And most of them, they are coming from the 1970s or 60s. They are not new discoveries of chemicals in the recent history of synthetic chemistry to act as nitrification inhibitors. I will tell you that industry is really eager for new molecules, but it seems that the synthesis of new molecules has stopped. So that's where we are with synthetic nitrification inhibitors. That what they do, we use them to inhibit the transformation of ammonium or ammonia to nitrite, and then this is changed to or transformed to nitrate. So synthetic nitrification inhibitors, how are they used? We usually co-formulate them with nitrogen fertilizers, and we call this mix stabilized fertilizer that we apply in soil, and we impose the slow transformation of ammonia that we apply in soil to nitrate. So we decelerate, in a sense, all this transformation process, and we reduce the consequences of releasing reactive nitrogen in the environment. How well synthetic nitrification inhibitors work today? Well, it's not an optimum option for our field. That's why we try to find other solutions. It's not that we don't like synthetic chemists. I mean, I work with pesticides my whole career, synthetic chemicals, but they seem to have a lot of consequences for our environment. So we seem to be able to find them a lot in water resources. So they contaminate water resources. They seem to enter the food chain, so we can find them in meat, in the meat of livestock, because they are uptaken by plants, and then the grazing animals take up synthetic nitrification inhibitors. And they also are not impressive in their performance, to be honest with you. They seem to have a particular selectivity on ammonia oxidizing bacteria, and they don't seem to do much with the other key player in the nitrification process, the ammonia oxidizing archaea. So they seem to have a rather narrow range of activity, but you know, it's acceptable. It was acceptable back then, because until 2005, where we only had synthetic nitrification inhibitors, we only knew that ammonia oxidizing bacteria are the only players in the nitrification process. They were working very well in AOB, in bacteria, so it was fine. But they don't do very well in ammonia oxidizing archaea. And the problem is that ammonia oxidizing archaea, they seem to be the key players in nitrification in acidic soils. And if you look at the soil map of Europe, there you have a lot of acidic soils. So you need to find a solution for these soils where synthetic nitrification inhibitors do not seem to be able to actually control the ammonia oxidizing archaea, that they are the key drivers of nitrification. So you need to complement maybe their activity, or to find other solutions, more sustainable solutions, to lead to decelerate the nitrification process. But to be absolutely fair, they do well in reducing N2O emissions. So if you look, I have put 3 different metadata analysis that they have actually came out in the last 4 years. So you can see they are generally able to reduce, in all different scenarios, the emissions of nitrous oxide by 40 to 50%. So they, after all, if you focus on the capacity to limit nitrous oxide emissions, they do well. Why they do well? They do well because nitrous oxide emissions are so much linked with ammonia oxidizing bacteria that they are the primary target. But let's see what plants can do for soil nitrification and how good chemists they are in inhibiting nitrification in soil. So it seems that it's the first observation of potential plants that they can inhibit nitrification goes back to the 1974. So it's not really an early observation. And then there were a lot of other papers coming up, but actually this whole area exploded in the last 10 years after the first true and clear verification that there are plants, wild grasses, that they are producing sex-specific lactones that they can inhibit nitrifiers in soil. And to be honest with you, I would say a rather funny story, because it was so much under our nose, all this story behind about biological nitrification inhibitors. We collaborate with a big agrochemical company, and they are asking us to actually screen a lot of different compounds. And we came up with to be testing. So they asked us to suggest some compounds. So we actually suggested some classic, very well-known compounds that they are produced by corn. And they were laughing because they had these molecules in the library for 30 years. And they hadn't realized that they act as nitrification inhibitors. And now in 2024, we're actually testing those compounds as nitrification inhibitors, while they were the pioneers in isolating them 30 years ago. So you understand that's, how much information was available, but has been overlooked. So which plants synthesize the plants that we know that they synthesize nitrification inhibitors? There are some cultivated plants, corn, rice, wheat. They produce stuff, but there are a lot of other ones that we don't know exactly why they actually inhibit nitrification. They exudate things, but we haven't isolated yet what is the key factor or what are the key chemicals that they inhibit nitrification. But for sorghum, for rice, for maize, for signal grass, we actually know very well what are the key players, what are the biological nitrification inhibitors that they inhibit nitrification, and they are produced by these plants. The biosynthesis of these chemicals by plants is really complicated. It's a very difficult puzzle, and you can see that the biosynthetic pathway of sorgoleon and the biosynthetic pathway of xenon, two known biological nitrification inhibitors from sorghum and corn plants. So really, plants under circumstances, they are excellent chemists. They can produce really good stuff. What do we do on this area in our lab? So we have been screening root exudates from our indigenous flora, and we are working with Trigonella. We screen for pure plant metabolites, and we also screen, along with our colleagues from University of Vienna, we are screening for BNI activity with exudates coming from wheat and pearl millet. And I'm talking about screening. How do we screen? So we actually have developed a fast-tracked high-throughput system, so that is composed of 3 ammonia-oxidizing bacteria and 2 ammonia-oxidizing archaea, that we can screen very quickly in 24 hours a lot of different extracts and compounds and get a sign of the activity or not activity in a high throughput level. And well, the question is why? Why plants produce nitrification inhibitors? There must be a reason why. So there are a couple of, I would say, obvious reasons why. Some might be random events. So there might actually might be an adaptation. for and conservation in soils that they are characterized by low nitrogen content, or they are just plants. For some plants, they might prefer to keep the nitrogen in the soil in the ammonium form because they actually prefer ammonium over nitrate. But it might be random as well. So from all these molecules that we are working on, I'm pretty sure that for some of these molecules, they are just allelopathic molecules, that they are produced by plants for other reasons, for allelopathy, to combat for space, for example. And it just happens that because they are very bioactive, to also inhibit nitrifiers that they are very sensitive, to be honest. Okay, so really this is not very clear yet why plants produce BNIs and under what circumstances, but they certainly do so. But how biological nitrification inhibition is applied in or can be applied in soils. So we notice, I mentioned earlier that chemical synthetic nitrification inhibitors, you just mix them with a nitrogen fertilizer and put them in the field. So here we have options. So one option that goes forward is to actually use either cultivated lines or plants that they are producing BNIs intercropping with cultivated plants. So we can actually use the plant as the source of continuously releasing BNIs in the soil and enriching the soil with biological nitrification inhibitors. But it will need an adjustment to our cropping systems, okay? Either intercropping with good cultivars, or maybe we will find good elite cultivars that can also produce biological nitrification inhibitors, and they can do both, producing well and producing also BNIs in the roots. And the other obvious way to apply is to find a way to produce these very active biological nitrification inhibitors that we have in our hands in very high amounts. And there you might be able to buy them from the market because they are already available, or you will need biotechnology or synthetic biology to actually create a plant-based system that it will actually produce copious amount and invest on the production of BNIs in soil. But how well they are doing, how well they work, biological nitrification inhibitors. In contrast to synthetic nitrification inhibitors, they seem to prefer to be more active against ammonia oxidizing archaea. So a good idea, mix them up. So use them because they seem to have complementary action. Synthetic nitrification inhibitors and biological nitrification inhibitors. One thought. They have a comparative performance with some known synthetic nitrification inhibitors at certain concentration levels, and as far as we know, most of the known ones, they degrade very rapidly in soil. So the question is that good or is that bad? We were complaining, I was complaining in my earlier slides that synthetic nitrification inhibitors, we can find them in the trophic system, or we can find them in water. And this is because they don't degrade very rapidly. Here, those are degrading rapidly. This comes with a cost, though, in a performance. So if you have a chemical that degrades rapidly, you lose performance, but you don't have any effects on soil health. Okay. However, consider that you have a plant there that is producing continuously, or we can regulate this in a time frame, and is producing that in the soil and enriching the soil. That might balance out the problems. So let's summarize. I promised I will give an Oscar. So let's summarize, okay? So synthetic nitrification inhibitors, well established in the market, mode of action not clarified, and this is something that I didn't mention. We actually hypothesize at the moment that they inhibit ammonia monoxygenase. However, we don't have hard evidence that this is how they work. We are talking about chemicals that they are in the market for 50 years now, and we still don't have the biochemical mode of action fully clarified. And this is something that actually Dimitrios is doing now. So we're trying to clarify the biochemical mode of action, the point and the mechanism of action of these compounds on ammonia oxidizers. They seem to prefer or to be more active on ammonia oxidizing bacteria. They have a lot of problems. They show erratic performance in soil. So depending on which soil we apply them, they work or they don't work, or the temperature is also affecting their efficacy. And certainly there are evidence that they contaminate natural water resources and they actually get into the food chain. So if we go to their to biological nitrification inhibitors, their opponents, they are not established in the market, so there is no single product in the market based on biological nitrification inhibitor, but we are trying to work on it, but we are not ready yet. Of course, the mode of action is not clarified, but again, we are trying to work on it as well. They seem to have a wider range of activity, but they seem to be, most of them, more active against ammonia oxidizing archaea. We don't really have a very good clear picture about their performance in soil, but in the field, but in soil, they seem to work well at concentrations that they are equivalent to what we put in with synthetic nitrification inhibitors. And certainly, they don't seem to have a potential to contaminate natural water resources, soils, or get into the food chain because they are not persistent. So the Oscar goes to plants. I was a bit biased. I had Professor Papadopoulou looking at me, what is he coming up with. So plants are actually they are the best chemists at the moment. So we're actually hoping that we will disentangle all the complexity of their chemistry and to identify molecules that they actually work well in soil against nitrifiers. And at the same time, they promise for a sustainable future in nitrogen cycling. So really, we hope that we will actually pass now, we are passing now in a new era or in the nitrification inhibition from the classic synthetic chemicals to the new era with biological nitrification inhibitors that they will be, I believe, will be coming soon in the market. So with this, I would like to thank you. And of course, I will be happy to answer questions. I will be around for the whole workshop and then in the pub. So if you have questions, throw it. Thanks. And from the audience, of course, if there are questions from the audience.
You agree with the awards?
Can we see the audience, the people that they might be linked? Questions? I've explained them very well. It's Panayoti.
Hi, I would like to ask a question. For the synthetic inhibitors, you said that they reduce the production of nitrous oxide. Do we know if something similar happens with the biological inhibitors?
Yeah, they seem to from the first data that we have, they seem to do well. But this you have to take into account that we don't have as many data at the moment as we have with ammonia oxidizing with synthetic nitrification inhibitors. But the first data that we have in our hands, they seem to indicate that they actually do equally well in reducing the emissions. There is a fly, isn't it? It goes to the entomologist back there. What is?
Thanks, Dimitri. Very nice talk. I want to ask do we have any information about these molecules coming from the plants? I mean, they produce naturally or under stress, or
yeah, we have first indications about it. They seem to, I mean, they seem to be produced under a high ammonium concentrations. Their production seems to be enhanced at the drought stress. So these are the first, I would say, hints that we have about their production. But also this, you have to consider that this depends on the plant most probably as well, how it responds to the different stressors, abiotic stressors. But these are the first ideas that we have. So we are actually trying with Trigonella now with to play around with the different conditions and see which are the ones that optimize the production of exudates that they have high nitrification activity, because we have in our first attempt we've got some really nice results. So.
Okay, thanks.
Who's that? Isn't it?
Yes, from abroad.
Yes. Yes. Who's that?
Dimitri,
Hi, Michalis. Okay.
what I would like to ask is, do you have any clues about the impact of potatoes? Do you know from literature if potatoes are producing BNIs?
No,
I'm
we
asking this
don't.
because we have very, very interesting results that you will see later on in my presentation, where the nitrous oxide emission is in a very intensive crop like potatoes are minimal. And we have very interesting results also in leaching, that we cannot find nitrates in the leachate. So from your presentation, maybe this is something that somebody should examine.
So you should examine it. But yeah, I mean, if we can look up in the soils uh, uh, for the levels of nitrifiers and see um, if they are really low, or if you have depending also on the pH and everything. So we can, I mean, this is interesting. So we can follow it up.
Yeah. Okay.
Okay. So, Hugo.
It's me.
Hi, Hugo.
Hi, Professor. First of all, thanks for the talk. It was very interesting. Nice results also.
Of course.
Yeah.
They are yours.
What I wanted to ask, and having an idea from the 3 talks until now, we have from Christa Schleper that one of the possibilities to reduce the consumption of meat and starting consumption vegetables. Graeme talked about the modification of the ecosystem or the microbes to improve this problem of the nitrogen, the reactive nitrogen. And now we are talking about plants and how they can also deal with this. So one of the major problem was the increase of fertilization to increase the mass or the production of the vegetable. And what do you think if we see it in a different perspective? Instead of increasing the fertilization, having crops with less fertilization, producing less fertilization. I believe that also the interaction of microbes and plants will be very important in this fight against.
So you mean to invest on crops or lines of crops that they will produce the same with lower nitrogen input? Yeah. Yeah, I think it will be a difficult task, to be honest with you, considering that this is a futile cycle at the moment, because growing population, using more fertilizer to feed this population, and blah, 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 and this goes on, and it actually magnifies. Um, so yeah, I mean, it is a ambitious idea. I'm not sure if Poppy has an idea about it.
We need new genotypes to achieve that. Okay, the genotypes that we were using now, they are selected to produce only under high fertilization and high water input. So we need to identify these specific genotypes that maybe there are out there, but we never looked for them. So elite genotypes that they also produce BNIs, for example, and use those ones in our cultivation.
Yes.
The other thing is that to identify genotypes that they do produce BNIs and then do accelerated breeding programs that we can do now. We can really bring in these traits in our elite genotypes and make new genotypes that we can use up there. So I say I teach in the 4th year of biotechnology, and I say that the solutions will come from biology. And so the Oscar definitely goes to the plants. So we just need to look deeper into this. And I would like to make one more comment. So we can choose which plants to look into. So we know for many of our plants that some of them, they are producers in secondary metabolites. And if we have some idea now which are the structures that we should look for, then we can go back in our available knowledge. We know the chemistry as from 1974, they knew which is the plant that can act as a nitrifier, and we can go back into available knowledge that we didn't look for and look deep well enough to identify which are the plant species that we can exploit better. There is a lot of untapped knowledge there and untapped chemistry that we never looked for and we can use.
Okay, so because we are running late, we will have
a short break,
yes.
Just 10 minutes. See you all back in 10 minutes then. So welcome back, everyone. I hope you enjoyed the short break. Next, we have Dr. Fotini Giannakopoulou, General Manager of the Hellenic Fertilizer Association. Fotini holds a PhD in soil science and has significant experience in both academic research and industrial innovation, focusing on sustainable fertilizer practices and policy development. Fotini, thank you for being here with us today. The floor is yours.
Hello, thank you very much. I'm trying to share my presentation. So just a moment, please. Everything is okay now? Do you see my presentation?
Yes.
Yes.
Great. Thank you very much for the invitation. It is really very interesting, and I'm going to feel a little bit pity not joining you in person. I think the next time I will be with you. I'm Fotini Giannakopoulou. As I mentioned, I'm an agronomist and I hold a PhD in soil science. And for the last 10 years, I'm the general manager of Hellenic Fertilizer Association. So, without any delay, as the key points for the fertilizer industry and the fertilizer production is already presented, I will start my presentation with the main challenges that the fertilizers industry faces in that period and a roadmap of what they are looking for. And I think that these ideas would be have a very good implementation in the industry. The Hellenic Fertilizer Association is a professional association. We have 73 members. They are companies that produce and distribute in the Greek market all type of fertilizers. And when I say all type of fertilizers, I mean mineral, organic, organomineral, and biosimilar fertilizer products. We have all the types of fertilizers, and our members all over Greece. As an association, we have a lot of partnerships. We are affiliated members with the National Fertilizer Association, national representative of the European Biostimulant Association, and our vision is to assist the Greek fertilizer sector with innovative research programs and interconnect all the stakeholders of agri-food chain. We have a lot of actions. You can find us in the social, but also in our website. So I won't spend any time to explain what we are doing. But we work with strategic partners. We work with many academic and research communities in order to serve the results of research programs, but also as we try to understand the market trends and what the industry wants in order to serve services also to the research program. The challenges that the fertilizer sector faces, I tried to summarize them. First of all, we have the food security. This is very important as the population is expected to grow. Fertilizer industry, I think it is mentioned before how important it is in order to give the food that the people needs. But we have to take into consideration the geopolitical shifts that we face these times. I think it's a very hard time for fertilizer industry. We know about the energy costs and how this had affected the recent years the prices of fertilizers. We have the environmental concerns. Top of all the agendas is to minimize the nutrient losses, and we will see in my next slides that this is very important for the industry. And of course, the new consumption habits, the health claims that the products should have. And this is very important because we have to connect the fertilizer industry with the requirements of the food industry. And of course, there are many regulatory frames, there are many European policies. We have to follow a very intensive regulatory frame. We have Green Deal with specific targets. Now we're waiting for the EU soil strategy, of course, the CAP. And I think in September, they published the strategic dialogue on the future of EU agriculture. And all these, they have a very direct effect in the way that the fertilizer industry acts. And of course, we have a new fertilizer regulation for how the fertilizers came to the market with very new requirements and specific data that the industry should follow in order to sell the products in the market, in the European market. I have some slides about this regulation, and I think it's very important, especially in order if you want to develop a new product, to follow the rules that the regulations ask. And we know, you know everything about the climatic crisis, how this affects the agricultural production. And here the fertilizer industry has new products that they are for many years under investigation from academic community and research community. I'm talking about biostimulants, but now I think they are used in the market more and more. So I just mentioned that the regulatory frame is very intensive. I have just selected some policies that we have to follow this period as an industry and to adapt and in order to be comply with what the EU wants. And I just, as I mentioned, the strategic dialogue that published in September was also has some main strategies that we have to follow. According to the farm to form strategy, the main reason that I put it is to mention that the goal of the farm to fork strategy is to diminish the nutrient losses by at least 50%. And it's very important because it's mentioned everywhere that to ensure no degradation of soil fertility. So this is a main goal that the industry should follow, and this I think that the use of nitrogen should be top of the agenda in all the fertilizer industry. And all this, if we as an industry decrease the nutrient losses, this will then reduce also the fertilizer use by at least 20% by 2030. Of course, there are other strategies, but this is the most important thing for an industry that should develop and apply relevant strategies to achieve this target. It is very important in order to have a transformation in the products that they produce and to use more friendly compounds. The strategic dialogue, we are waiting to see exactly how all these principles that they agreed by all the participants will be implemented and what strategies will be focused. I only mention that the three words that they use, it's how important is the agriculture, the sustainability of agriculture, but also the resilience of the systems. So if we want to summarize, the European strategy, which was announced in these recent years, promotes the transition towards a sustainable food system, setting new targets for all actors of food agri-food chain. So all the distribution that we have with the biodynamic, with an energy and climatic crisis, as well as with the political uncertainties emerge how important is the agri-food sector and the fertilizer industry. We know about the climate change and how the EU farmers have to adapt these new challenges and the regulations. We have to see new perspectives in agriculture inputs, especially in our fertilizer product sector, in order to follow these climate changes and these abiotic stresses that we face. According to this research, and this is something that our industry very often discuss, is that abiotic stresses are the main cause for crop losses. With the green color, you can see the actual average yield, and with the red color, you can see how bigger could be the yields if we haven't these losses caused by abiotic stresses such as extreme temperature, drought, lack of oxygen from water loading, and salinity. And this is something that we have to focus on it. We have to find solution in order to provide new products, fertilizer products to our farmers. According to the new fertilizer regulation, when we say fertilizer products, it is a big part of it contains a lot of, it contains not the classic fertilizers that we use, it contains also biostimulants, also the inhibitors. So altogether we have to find new products that we will address in order to minimize the effects of abiotic stress. Some Inside information for the fertilizer sector is today we don't just cultivate plants, we manage ecosystems, and the main theme that the industry should focus is to produce more with less. And according to this diagram, we are trying to promote the integrated soil management, not use only mineral fertilizers, because in Greece, as in many other member states, the consumption of fertilizers as well as the nitrogen units has a significant fluctuation per year. Especially in Greece, they present a downward trend. Inorganic fertilizers, although continue to serve as the primary nutrient source, of course, of crops, with compound fertilizers being the preferred choice of our farmers. So nitrogen management in soil plant system, we have seen that via inhibited fertilizers and slow release fertilizers significantly increased in recent years. Plant biostimulants also, especially humic and amino acids products, have been integrated into the producer's soil nutrient management. And here we have to say that plant biostimulants are a category, are an umbrella that contains a lot of different products that are expected to co-evolve with mineral and organic fertilizers. And we have a very intensive fertilizer regulatory outlook. As you see, we have to follow the EU fertilizer regulation, which came into implementation in 2022. It is a regulation that is an optional harmonization. This means that they are laying down rules of making available on the market EU fertilizer products which have CE mark, but it's very important to say that the member states could have their own, they could have and comply with their own national fertilizer regulations. And as in all other member states, in Greece, we have our national fertilizer regulation, and which have specific requirements and in order to register in Greek market. 2 slides for the new EU regulation, it's CE mark fertilizer products, and in order to have a PFC, to have a fertilizer, it should belong in one of product fertilizer category. We have 7, as you see. One of this category is the number 5 is inhibitors, and in order to have a CE fertilizer, should have one of the 70 component. There's a specific component material, and only in this case, you have a producer can mark the CE fertilizer. And for the first time in this regulation, we have a common and legal definition for plant biostimulants. Plant biostimulants, according to the EU fertilizer regulation, means a product stimulating plant nutrition process independently of the product's nutrient content, with the sole aim of improving one or more of the following characteristics of the plant or plant resource. And there's these 4 claims that the producer should make experiments in order to prove that they increase the nutrient use efficiency or the tolerance to abiotic stress, quality traits, availability of the nutrients in soil or resource. And it's very important because we have the technical specification in order to prove these claims. So for the first time, plant biostimulants is recognized as in order to do the claims and not the compound that exist in order to be a biostimulant. What the fertilizer industry is doing. As I said, I think that the fertilizer industry is transforming in these days. The fertilizing products should be focused on improving the nutrient use efficiency, and there's three different directions that we see in fertilizer industry. First of all, we have fertilizer products for nitrogen management, and this is very important, as we mentioned, also for the environment. In this period, these products is also included in the CAP, and they gain some extra amount of money if the farmers use these products in the ecosystem, in the ecosystems, sorry, I'm sorry. And we have nitrifications from synthetic inhibitors, as mentioned. According to my knowledge, I don't know, and I think that the industry doesn't use plant inhibitors. And also we have for nitrogen management, the slow released fertilizers, which are coated and use specific polymers. The second product that very focused this period, the fertilizer industry, is the biostimulants, microbial biostimulants that is recognized by the EU fertilizer regulation, are the four categories that you can see in my presentation. And the non-microbial, we have put some components that they used as a non-microbial, but as I mentioned, this is based on claims that the fertilizer producer wants or have proved according to the experiments. Of course, the biostimulants can be sold in the market with national regulation, as I mentioned, with the specific rules that national regulations has. And the third pillar that the fertilizer industry these days is focused is to produce low carbon fertilizers. And the low carbon fertilizers, they're again two different directions. First of all, is in the production stage. There are many investments for the blue ammonia and green ammonia, and also in the EU, the decarbonization of fertilizer industry, as we know how intensive is the need of energy. There are many, I think they tried hard, but there are many problems that we face in this industry for this direction. And the second is to produce low carbon fertilizers in the meaning of the uses, in the uses of fertilizer in the farm application, and in the technologies that they use in order to minimize the carbon footprint of the use of fertilizers. So, as trying, I think the main pillars that the fertilizer sector is focusing these days is in the integrated nutrient management. Of course, they are applying the 4Rs principle, the right source, the right rate, the right type, and the right place. It's the precision agriculture that they use for environmental, of course, specific benefits. The use of fertilizer products that improve the nutrient use efficiency. It's very important to minimize the losses of nutrients, and of course, they add in their portfolio and their solution, the digital solution, digital apps that they will use in order to specific use the fertilizer products in specific crops. And all these, they have environmental benefits, especially for protection of the water quality, the improving the soil fertility and minimizing the carbon footprint. And also, I would like to add that as we see, the regulatory frame is very intensive, and now is under evaluation also the nitrogen regulation. So we are waiting to see what new is going to have to follow according to the nitrogen management. With these techniques, also, except from agronomic, from environmental benefits, the fertilizer industry focuses and aims to some agronomic benefits. They want to enhance the crop yield potential, to reduce the nutrient losses, and to increase the plant tolerance to abiotic stress. Of course, they want to increase the crop yields, the quality products, and the nutritional value of the foods without increasing the use of fertilizers. So if I want to conclude, I think this period is very important, your project, because there is an ongoing transition of fertilizer industry, focusing on knowledge, innovation, development, and synergies. Today, the plant nutrition sector is betting on the developing of tailor-made solution products and appropriate to each crop and region. And this is a very high bet, I think, and very difficult. But there's a lot of things to research and to develop. Also, the plant nutrient sector is on the developing of integrated soil nutrient management and products that they increase the nutrient use efficiency. Of course, the implementation in the farms of these products requires specific knowledge and continuously technical support at the plot and the crop level. And this is very important and very difficult. And the most important thing they want as an industry to add value to the whole agri-food sector. So we think that joint forces could help to make a positive impact also to the environment, but to the farmers. Thank you very much for your attention and for the invitation to join this workshop.
Fotini, thank you very much for this informative talk. It was really nice to hear all this data together, all this info together. Are there any questions to Fotini?
Hello, I'm Alex. Thank you very much as well for the very nice talk, very informative. Should we give priority to Graeme? Okay. So I have two questions, if I may. Sorry, Graeme. The first question is that, according to my knowledge, when we talk about nitrogen in fertilizers, nitrogen can be in the form of either urea, ammonia, or in some cases nitrate. And based on all the talks that we heard today, that nitrate can be leaching from soil actually and not be that available to plants. Are nitrate fertilizers still preferred in the market by farmers?
No. As I mentioned, in Greece, but also in most places in the other EU member states, compound fertilizers is the common form that the farmers use. Especially with inhibited last years, we can see that inhibited fertilizers increased, but and especially the nitrogen use it has been diminished in the last 3 years very much. In Greece, although we see that for many years we face an under fertilization. The use of nitrogen is almost 60% lower than they used to be in previous decades. And I think this is a tension, the trend in all over Europe, but especially in Greece, due to the crisis that we have, the economic crisis. And I think that also our farmers have changed some traditional method and applications. Of course, there is a lot that we should focus on. We have a lot of problems. We have to educate the farmers to make them to understand how valuable it is to change their cultivation practices.
Okay, thank you for that. And the second question actually it refers to nitrification inhibitors, because you talked about the change in policy making for the European Union, the regulation, and you touched a bit on nitrification inhibitors that they can be classified as CE fertilizers if they have some, they fulfill some parameters. Can you expand a bit on that?
Yes, according to the new fertilizer regulation, it's very important to understand that the EU regulation sets specific requirements. One of these requirements is to have some experiments in order to, they're not compounds in this regulation. They're not like the previous EU regulation that we have the MTP, DCT, things like that. Does not exist in this regulation. We don't have compound specific. We have only requirements, only new inhibitors should fulfill the requirement, for example, to have 14 days to reduce the nitrogen losses up to 20%, or I'm not sure, I can't remember exactly the requirement, but I can send you the this EU regulation in order to see exactly what we have to follow.
Okay, thank you very much.
Sorry for that. I will find it exactly to find. I think it's 14 days.
Yes, thank you very much for that excellent talk. It's very informative. The question is maybe quite specific, but it's about the coated slow-release fertilizers. And I was wondering, is there a much greater cost associated with these slightly more, I don't know, sophisticated is the right word, but these slightly more formulated fertilizers compared to, say, ammonium nitrate? And is that does that prevent uptake by farmers?
Both of the fertilizers, it's a very good question. Yes, really, it's a little bit sophisticated, all these products, because you have to educate the farmers, and it's difficult for the producers also to produce and to experiment and to find all these products, especially in the fertilizer industry. That's why I put it in different as the products that they are focused on nitrogen management. Although the inhibited fertilizer is something very specific with the inhibitors. The slow-release fertilizers, usually they have as a compound polymers, or they have synthetic other materials. And I think there's a lot of space for research and development, new products in that area. It's a little bit, but they use it as a primers in farmers, in the farming application. Okay. I'm not sure if this is okay for you. Sometimes all this, it's very.
That answered it perfectly.
This regulation is very new for the industry, and until now we have 14 modifications. So especially for polymers, the last modification and the criteria in order to be that they will make a slow release fertilizers published last week. So it's a living document, this regulation. It's very new products to specify exactly how the producers will follow the requirements.
Okay, thank you, Fotini. Thank you, Fotini, for your insights.
Thank you very much for the invitation.
So finally, we have Dr. Michalis Somirou, senior researcher at the Agricultural Research Institute in Cyprus. His work focuses on soil microbial ecology and mitigating greenhouse gas emissions from agricultural systems, particularly in semi-arid environments. Michalis, we are looking forward to your presentation.
So good evening, everybody. I will try to share my screen. Do you hear me?
Yes, Michal, we can hear you.
Let me. Can you see my presentation? Because I cannot see if you...
Yes, we can see your presentation. Go ahead.
Okay. Good evening, everybody. Thank you, Dimitri, for the invitation. It would be a pleasure to be there, but unfortunately, I couldn't manage to do this. I will try to keep my presentation for 20 minutes. I will make an introduction. I will present the greenhouse gas emission in Cyprus. I will present the inventories and methods that we have to follow. We'll present the infrastructures that we have in ARI for measuring all these methods. And I will present 2 case studies, one for potato, one for manure, and then I will conclude with some main messages. So EU has a very ambitious program through the European Green Deal. So the economy of European Union should be net zero in terms of greenhouse gas emissions. And this means that we have to make deep emission cuts across all sectors. And what I would like to stress here, and this is something that it's my strong personal view, is that agriculture is not the main emitter. So we have to disaggregate all this wrong perspective that agriculture is a main emitter. So for Cyprus, for example, agriculture since 2005 was participating around the almost 6%, and today is around 6.6% of the total emissions. And we have, I don't know for Greece, what will be the target. Until 2030, we have to reduce the emissions that are not included in the ESR scheme. ESR means that is a penalty that the consumers are paying for the consumption of electricity that is translated in the emissions that the gas participates in the global warming potential. So for agriculture, for Cyprus, we have to reduce more, around 32% of the emissions that we are already emitting. So this is a huge number, and we have to find solutions how this will happen. So what is the current methodology and how we calculate all these emissions in soils and particularly in soils? In Cyprus, we are using the tier 1 method. This is the simplified method for estimating the nitrous oxide emissions. It uses the globally average default emission factor, and actually it uses this very simple equation, let's say, and the default value for the countries that do not have direct measurements from their ecosystems is 1%. This means that every 100 kilograms of nitrogen that is imported in the country, the 1% is calculated as nitrous oxide emissions. So this was the situation for Cyprus during 2021 before we revised our emissions after our group research in field experiments. And this is translated to almost 5 million euros per year for Cyprus. And you can imagine the amount of money that could be, and also the baseline emission trend that the countries, especially the Mediterranean countries, will reduce compared to the temperate regions of European Union. And through our research, the revised new emission factor will be reduced. This is a tentative figure to 0.15%, which means even more even higher reductions in the soil. So this is a very nice figure for for the current situation in Cyprus. So before the revision of our national emission factors, the contribution of soils, mainly the nitrous oxide emission, was the 19% of the total greenhouse gas emissions of the country. And after the field measurements and all the research that we did, the contribution of soils, the real measurements accounts for 9%. And we can see here that the enteric fermentation and the manure management are actually the main emitters in our agricultural ecosystems, with the cost of enteric fermentation to be almost 24 million euros as actual cost for our economy. And this, of course, is related with the number of dairy cattle that are raised in livestock industry of the country. So how do we monitor all this around 2016? And here is our first publication for the effects of chemical and organic nitrogen inputs on nitrous oxide emissions. We did all this work with static chambers and we didn't measure any productivity of the crop. We didn't measure the carbon sequestration with custom-made chambers and with manual sampling in the field. And I can assure you that I will not do this again because it's very difficult. The measurements are not so accurate compared to what you will see later on. And despite this, we've seen that in our systems, in the rain-fed systems, the emission factors was only 0.33%. And what we found in our experiment is that the main factors in rain-fed systems in our soils, which are alkaline soils, is nitrogen availability and water availability. So these are the two main factors that are controlling the nitrous oxide emissions in our agricultural ecosystems. Now here is the experimental setup, some photos from our experimental station with the potato crops. We have 2 consecutive years experiments running out there, the 2023 and 2024. And I will present you the results from 2024. And here we can see that after the nitrogen application, we have an increase of nitrous oxide fluxes, which is expected. But this is clearly related with the plant growth stage of the crop. And here we have the control and the treatment with the nitrogen fertilization. And the take-home message here is that the emissions factor is much lower than it was not expected. So we have applied nitrogen according to the best agricultural practices that the farmers are implementing in our systems, and we have seen that the emission factor is actually very low. But this was not the case for ammonia. And here we can see that during the second application, we had a peak of ammonia within our system. And this is probably related with some environmental conditions that took place. If there was a raining day with high temperatures, which means that the ammonia was favored under these conditions. And also, we found that the emission factor of ammonia was also very low. It was only 0.1%. When you add all the emissions, I didn't show you the emissions. I have them in the slides later on, but I will not present it for the sake of time. If you see the budget of the global warming potential, the carbon dioxide, the methane, and the nitrous oxide, we can see that the potato crop, a very intensive crop in Cyprus, is absorbs. It's not does not contribute to the global warming potential. And this is a very important take-home message for our ecosystems, because we are implementing fertilizers. If a farmer implements a fertilization strategy in a proper way, then the warming potential of his crops is negative. So it acts as a sequestrates carbon dioxide equivalents. Now let's go to the manure now. For nitrogen and dairy cows, we've seen that we have an X-ratio, an intake of more or less of 700 grams per day per dairy cow, and there is a huge amount of nitrogen that is excreted through urine and feces. And for the dairy cow, we have this, let's say, pathway. We have the organic nitrogen, the urea, that we have the ammonia emissions, we have the ammonium transformation, nitrates. Graeme and all of you, of the speakers below, show this pathway. So from the stockpiles, we have emissions of nitrous oxide and ammonia. Within the barn, we have, this is a completely different story, and it's very sophisticated experimentation in order to calculate the actual emissions that you have within the barn, especially in our cases where the barns actually are not closed, are open. So what we did, we have a completely randomized design experiment. We have dairy cow manure and goat manure during the summer. We have a completely dry manure and we simulate a raining event that, due to climate change, we have some days during summer in Cyprus that we have heavy rain events during summer. We did a lot of measurements, but today I will present only those that are related with the greenhouse gas emissions. And here we can see again, what I would like to show here is about the contribution of nitrous oxide emissions in the total greenhouse global warming potential of the manure stockpile. Another thing that I would like to show here is that during the dry period, the emissions were, the fluxes were zero, were minimal, except the first day of the initiation of the experiment because of the disturbance of the manure. Once we have the raining event, the addition of water within our system, we have a peak of the emissions, and these were mainly derived from the heterotrophic respiration of microbes. And here is the contribution of each greenhouse gas in the global warming potential. And we can see the difference between the origin of the manure, and what is very surprisingly here is the consumption of methane. But this is something that is out of the scope of our presentation today. For the ammonia now, we can see here the fluxes. What is important to stress here is that in these two, these are the DL flux figures of the ammonia. The gray column represents the dark hours of the night, while the white strips represent the daily hours, and we can see that ammonia follows actually the photosynthetic rate of the crop. We have peaks during the day, and again we have here after raining a peak again that was higher in the goat compared to the dairy cow. And we asked the question which factor was critical, and we've seen that in both cases there was a significant positive relationship between the ammonia in the manure with the ammonium in the manure with the ammonia that was emitted. Structural equation and modeling shows also that ammonia oxidizing bacteria were significant for the nitrification, and we have a very positive and highly significant relationship between the ammonium in the substrate with the two gases that were evolved. Here we have the population, the gene copy numbers as a relative to the total bacterial community in both cow and goat, and we've seen that here after the addition of water, we have an increase in the population of ammonia oxidizing bacteria. So, as a conclusion, we've seen in our experiments for almost a decade now that the nitrous oxide emissions are limited and they are substantially lower from the default values set by the IPCC. And this is extremely important for our competent authorities. They need to fund infrastructures that are able to perform measurements and perform research in an accurate way in order to provide them with the scientific valuable data that will set up the real situation of the emissions. Okay, so the measurement methodology is critical for the accurate estimation of the greenhouse gas emissions. This is something that we also realize with the static chambers and the manual handling of the gas sampling and things like that. The overall global warming potential of nitrous oxide emissions in Cyprus is negligible, and it is similar with that of ammonia. And the presence of crop must be included in the field trials that will be used as reference for the inventors. So crop has a significant effect regarding the emissions. We have seen this also in methane, but also in nitrous oxide. And this was a question that I did to Dimitris before about how BNIs could be involved in these emissions, because in our system, we can measure the emissions actually within in a setup that includes the crop. So for the manure, the origin affects the greenhouse gas emissions. In our case, goat manure are higher compared to dairy cow manure. A precipitation event during summer will boost the microbial activity. This is expected. So shading could be a significant policy measure in stockpile facilities in order to avoid this kind of emissions. The amount of ammonium in the manure is a key nutrient and is associated with emissions of nitrous oxide and ammonia. And of course, the ammonia oxidizing bacteria is a critical guild that is associated with nitrous oxide emissions. I would like to thank all of you for your presence here, and also my team that without them I couldn't present all this data today. Thank you very much.
Michali, for sharing all your interesting findings with us. Are there any questions to Michali?
Hello. Thank you for delivering us this informative presentation. I would like to comment about the heating capacity between CO2 and nitrous oxides.
Yes. What is the question actually?
To compare, to comment all these results.
The equivalent of 1 gram or 1 kilo of nitrous oxide, the thermal capacity of the nitrous oxide is 265 times more the carbon dioxide. But the figure that I showed before is actually after the transformation to carbon dioxide equivalents, okay?
That was the point. Yes, thank you.
So, Michali, can you give us your presentation so we will go for asking money here in the region, in the prefecture of Thessaly to start off with?
Yes, of course.
Okay. So I'll pay your travel to come over here and we go together to ask for money.
By all means, Dimitri, because we do not. I was in a meeting in the ministry a week ago, and what we are stressing is that we have to measure what we actually emit. Okay, this is fundamental. All the IPCC regulation and all the stuff globally was based on a publication of Bauman back in 2000, I don't know, 1992.
The question is, if all this money that they are saving now from reducing the emission factor, are they coming back into your lab?
No. No, but what is important is
not even a fraction, not even a fraction.
No, we have now apply for creating a national unit, research unit for monitoring and combating mitigate the greenhouse gas emissions. So we ask for money, and I think they will give it to us because we have a lot to do for the manure and the livestock.
The question is, how much does it cost to set up the full setup for the chambers and the online measurement of such a unit?
We got a grant, a competitive grant in our institute it was funded by the government, and all the stuff we have in the lab now in our center is 2.5 million euros.
No, I'm talking about just the infrastructure, like the
infrastructure. We have 6 picaros and 70 chambers. It's almost 1.5 million.
Okay, so the 70 chambers with the 6 picaros costs 1.5.
Yes, with the green feeds, with the 2 for the animals also include.
Okay. So I'm getting some trousers with big pockets, and I will go to the region with your presentation.
Yeah.
Okay.
I can. I think if they realize, I don't know what are the targets of the Greek Department of Environment are for the 2030. I think Cyprus is the target. We are not going to get the target of the 32% reduction, and we have to pay a lot of money. So they have to reduce the overall cost of the emissions in order to pay less, to buy the rights of, because as
but I don't,
Christina
Greece,
said before, the Denmark reduced the emissions from ammonia 70%, and this reduction provide to Denmark a surplus of savings. So for Cyprus, Greece, Italy, maybe we will go and buy rights from Denmark. So we'll pay the Irish, the Danish, the Northern European, sorry, Grant, for that, for not doing what we have to do in our country. So simple.
No, but yeah, that's a strong argument. But that's a strong argument for the government. But I don't think anybody in Greece at the moment, as far as I know, they are measures systematically and monitor systematically the emissions, at least from agriculture. Okay, so I think that's a great opportunity.
There is a live project, the MedClima. Benagio was lead of it. They tried to do something with some LiDAR devices, but I don't think that they do.
Okay.
They are far away from measuring real measurements.
Okay. So thank you, Michalis, very much for a really nice presentation. Excellent results. We are running late, so we will close with your presentation now, because we have to carry on, and we have to go to the pub now. So unfortunately, you are not here, so you will miss out.
Okay, so, you can drink a beer for me.
I'm pretty sure that Graeme will have a major issue with that, but I will see you in a couple of days in Lyon, mate.
Yes, you shall. You shall. You're the first round.
Okay, so thank you all for being around, for giving the talks. And we'll keep in contact for anything else that might come up. Okay. See you then. Bye.
Bye.
Ciao.
Bye.